Join the Comgiverse Patreon for exclusive content, behind-the-scenes access, and special perks. Support the channel and connect with a community of revolutionary intellectuals and scholars. Click the link in the description to join now.
My name is Ed Green, and I am an assistant professor of biomolecular engineering at University of California, Santa Cruz. And my research program is around genome sciences. We study all kinds of interesting biological and evolutionary phenomena by sequencing, assembling, and analyzing genomes. We sequenced the genome of the Neanderthal, made a draft sequence of this back in 2010, and more recently, Svante's group and a big consortium of folks have published a finished genome, a finished quality genome of the Neanderthal. This has been an incredibly useful reagent for understanding human evolutionary history in many ways. First of all, it lets us know how we were related to them in great genetic detail, something that was really hard to do by only looking at the fossil and lithic record, just looking at the bones of our ancestors and their ancestors and the tools that they made. Opinions varied greatly on how much interaction there was between them and us in the past, and now we know that there was at least some interaction, and at least some interaction that resulted in viable and fertile offspring hybrid individuals, because we see the genomes of, or the genes of Neanderthals in many people throughout the world today, people whose ancestry goes back anywhere in Eurasia and even the New World, have a few percent of their genes came not from this group of people in Africa that evolved into humans, but instead to the group of people who were living at the same time in Eurasia, the Neanderthals, who are known from the fossil record. We got DNA out of their bones, and we see that that DNA can be found in people today.
You did an interview recently where you talked about some of the, the racist things that people have said about black people and monkeys and so forth. But you said when you actually scientifically look at white people, monkeys are hairy. White people are the hairiest people
on Earth.
On Earth. I am really hairy. Like, no, seriously.
I don't shave.
No, I shave all. I'm shaving
right
now.
I
got hair on my back.
Coming up
my chest is all hairy.
The hair coming up.
Hair on my ears.
Collar. Okay.
These
were highly overlooked similarities.
Yeah. The big ears. White people usually have big ears.
Bigger ears around.
Monkeys have thin lips. White people, I have thin lips.
Yes.
So really, I look more like a monkey than anyone else. But then there's the whole racist stereotype that to this day still goes.
The 19th century anthropologist is probably the most racist period in the most racist branch of science there ever was.
A new look at our linguistic roots. Linguists and archaeologists have argued for decades about where and when the first Indo-European languages were spoken, and what kind of lives those first speakers led. A controversial new analytic technique offers a fresh answer. Almost half of all people in the world today speak an Indo-European language, one whose origins go back thousands of years to a single mother tongue. Languages as different as English, Russian, Hindustani, Latin, and Sanskrit can all be traced back to this ancestral language. Over the last couple of 100 years, linguists have figured out a lot about that first Indo-European language, including many of the words it used and some of the grammatical rules that governed it. Along the way, they've come up with theories about who its original speakers were, where and how they lived, and how the language spread so widely. Most linguists think that those speakers were nomadic herders who lived on the steppes of Ukraine and Western Russia about 6,000 years ago. Yet a minority put the origin 2,000 to 3,000 years before that, with a community of farmers in Anatolia, in the area of modern-day Turkey. Now, a new analysis, using techniques borrowed from evolutionary biology, has come down in favor of the latter, albeit with an important later role for the steppes. The computational technique used in the new analysis is hotly disputed among linguists, but its proponents say it promises to bring more quantitative rigor to the field and could possibly push key dates further into the past, much as radiocarbon dating did in the field of archaeology. I think that linguistics might be in for a sort of equivalent of the radiocarbon revolution, says Paul Hegarty, a historical linguist at the Pontificia Universidad Católica del Perú in Lima, and a co-author of the new study. He described the computational approach in the 2021 Annual Review of Linguistics, revealing dead languages. To understand what's going on, it helps to look at how the study of Indo-European languages developed. During the 16th century, as travel and trade put Europeans in touch with more foreign languages, scholars became increasingly interested in how languages related to one another and where they might have originated. In the late 18th century, Sir William Jones, a British judge in India, noticed similarities in vocabulary and grammar in Sanskrit, Latin, and Greek that couldn't have been coincidental. Historical linguists have reconstructed much of the grammar and vocabulary of the ancestor to Indo-European languages, to the point where we can piece together what conversations might have sounded like. Turn on closed captions to see a translation of the reconstruction presented here. For instance, the English word father is pita in Sanskrit and is pater in Latin and Greek. Brother is brata in Sanskrit, frater in Latin. Although Jones wasn't actually the first to notice the similarities, his pronouncement that there must be a common origin helped to spur on a movement to compare languages and trace their relationships. A major advance came in 1882, when Jacob Grimm formulated what would later be called Grimm's Law. Grimm is best known today as one half of the brothers Grimm, who collected and published Grimm's fairy tales. But in addition to being a folklorist, Jacob Grimm was also an important linguist. Grimm showed that as languages developed, sounds changed in regular ways that could help make sense of how languages were related. For instance, the Indo-European word for two was duo, but duo was one of a number of words whose initial D changed to T as it passed into the common ancestor of English and German. Later, the T sound became Tis in an ancestor to modern German. So the Indo-European word duo became two in English and zwei, pronounced tiswai in modern German. Other words starting with the D sound behaved similarly. Scholars discovered a lot of these sound shift patterns, each obeying different rules as one language gave birth to another. Together with these sound shifts, linguists also study how words are formed, such as the way that English adds an S to make a word plural. They also look at how words are arranged, such as the way that English puts subjects before verbs and verbs before objects. And of course, they look at shared vocabulary. By comparing all these features of different languages, linguists are able to map how languages descended from one another and to place them in family trees that show their relationships. A chart illustrating Grimm's Law through differences between French and English pronunciation of related words, such as trois and three. Grimm's Law describes the regularity of how sounds change in languages. The chart shows how some sounds from Proto-Indo-European shifted in Germanic languages, such as English, while remaining the same in non-Germanic languages, such as French. Today, linguists are in broad agreement on the basics of Indo-European language groupings and how they are related to one another. They agree that the original language, which they call Proto-Indo-European, split into 10 or 11 main branches, 2 of which are now extinct. They also generally agree on where to put languages within the main branches. For instance, they know that the Italic branch gave us Latin, which itself developed into the Romance languages, such as French, Spanish, and Italian. The Germanic branch developed into languages including German, Dutch, and English. And the Indo-Iranian branch resulted in languages like Hindi, Bengali, Persian, and Kurdish. Ancestral lifestyles. By tracing changes in language backwards towards their sources, linguists have deduced many of the basic characteristics of the original Proto-Indo-European language, including some vocabulary, how words were formed, and some idea of how they were pronounced. And many linguists think they have even found hints of how the first Proto-Indo-Europeans might have lived. For example, the Proto-Indo-European language had a word for axle, 2 words for wheel, a word for harness pole, and a verb that meant to transport by vehicle. Archaeologists know that wheel and axle technology was invented about 6,000 years ago, which suggests that Proto-Indo-European can't be any older than that. If it was older, in other words, if it had started to split into other languages before it had words for axles and harness poles, then its daughter languages would have had to invent their own words for these things. The fact that they used the same words suggests that the split started after these technologies were developed. Other words in the language suggest that the first Indo-European speakers were probably familiar with horses, cattle and sheep herding, dairy, wool, honey, and mead. They seem to have had chiefs. The word reg gave us our English word regal, and may have been patriarchal. They had words for in-laws that applied only to the bride's side of the family, suggesting that the husband's family was considered primary. Many linguists think the vocabulary paints a picture of pastoralists, nomadic herders, who used horses and wagons, combined with genetic evidence that people dispersed rapidly out of the steppes into Central Europe about 5,000 years ago. They conclude that Indo-European languages moved out of the steppes and spread with the pastoralists. According to one theory, Indo-European languages might have been spread by pastoralists traveling in wagons like this early Bronze Age copper model from Anatolia. In 1987, though, the Cambridge archaeologist Colin Renfrew rejected a pastoralist origin for Indo-European. Renfrew reasoned that the dramatic spread of Indo-European languages must have required a bigger push that could be provided by contact with ragtag groups of nomadic herders. For a major shift in which a single language grew to dominate a region stretching from Ireland to India, Renfrew argued, you needed a more powerful force. He found it in the spread of farming. Simply put, as people took up farming, their population grew more quickly than that of their hunting and gathering neighbors. As farming expanded, the languages moved with it. Archaeological evidence shows that farming had begun moving out of Anatolia about 3,000 years earlier than the spread of pastoralists out of the steppe. So, Renfrew concluded, farmers were the real force behind the spread of Indo-European. By the time the pastoralists started migrating, the farmers they met were already speaking an Indo-European language. Renfrew largely dismissed the linguistic reasoning that the steppe hypothesis was based on. The commonality of words for wheel, wagon pole, and the like, he said, can be explained by parallel shifts in which different languages draw on the same base meaning when creating a new word. For instance, the original meaning of the Proto-Indo-European word for wheel seems to have meant something like circle or turn. Different languages might have inherited that basic meaning and drawn on it independently when creating their own words for wheel. Likewise, if the word thil for wagon pole had a more general meaning of stick or pole, it could have been adopted to mean wagon pole by more than one language. Searching for rigor. Arguments like these led a few linguists to try a more quantitative approach to reconstructing the history of Indo-European. For this, they borrowed a technique often used in biology to build evolutionary trees based on measurable traits. Their approach, called computational phylogenetics, treats languages as evolving systems, similar to biological organisms. But instead of tracing changes in DNA, as computational phylogenetics in biology does, the technique in linguistics traces words. Specifically, most analyses have looked at patterns in words that mean the same thing in different languages, and that can be traced back to the same Proto-Indo-European root. The more similar those patterns are, the more closely related languages are generally thought to be. While this may sound like the language trees long used by linguists, the trees produced by computational phylogenetics are far less subjective. The method is governed by strict algorithms and explicitly stated rules. In a sense, the computer program works by drawing a language tree and estimating the probability that it is correct given all the data and assumptions. Then the program makes a single change to that tree and compares the probability scores, keeping whichever tree is more probable. The process is repeated, sometimes millions of times, resulting in a set of most probable trees. These trees show how closely related languages are to one another. To estimate timings, when languages originated and diverged from one another, the researchers also provide the computer program with dates for when they think different languages existed, based on the best estimates of experts. Latin, for instance, existed around 2,050 years ago, Old Icelandic about 800 years ago, and Mycenaean Greek about 3,350 years ago. The computer program uses these anchor dates to create its timing estimates, including a date for the ultimate origin of Indo-European. The results can be combined with the historical record of where languages were spoken to help figure out a likely map of how they spread geographically. And the dates can be combined with the archaeological record and studies of ancient human DNA to see if the Indo-European language lines up with an early farming origin or a later steppe origin. Contradictory results. One such analysis, published in 2012, pointed to an origin of Indo-European about 9,000 years ago in Anatolia, supporting the theory that Indo-European originated with farmers. But just 3 years later, a different team used much the same data to conclude that the origin was just 6,000 years ago on the steppes, supporting the opposite view that pastoralists were the first Indo-European speakers. How could the two teams reach such different conclusions from such a similar list of words? A map of Europe and Asia illustrated two possible origins of Indo-European languages. Most historical linguists favor the origin illustrated in the top map, where the languages originated in the steppes about 6,000 years ago. A minority favor an origin among farmers about 9,000 years ago. Hegarty delved into the problem and discovered that the issue lay with the dataset used for both of these earlier analyses, which was largely based on one originally put together in the 1960s by Isidore Dayen, a linguist at Yale University. Dayen's dataset had not been a problem for the research Dayen was doing, but when used for the new computational technique, it was throwing off the findings. Computational phylogeny works best when there is a single word for every root, meaning researchers are interested in tracing. But the meaning dirty, for instance, can have a number of synonyms in English, including filthy and unclean. The Dayen dataset included synonyms like these for some words in some languages, but not for others. Including any synonyms at all, Hegarty realized, made the dataset harder for the new computational technique to use. But having an inconsistent number of synonyms, more for some languages, fewer for others, really threw the calculations off. I said, look, we have got to do this database completely again, from scratch. We have got to do much better, Hegarty says. So he and his colleagues chose 170 core meanings they wanted to trace. Basic words you would expect languages to preserve, such as words for counting numbers, body parts, colors, and things like house, mountain, laugh, and night. Then they brought together a team of more than 80 linguists and had them determine, for each of 161 Indo-European languages, the primary word for each concept. Only that word, and none of the synonyms, went into the analysis. We made a highly consistent database out of it, in a way that nobody has ever done before, Hegarty says. And we did a lot of analysis to make sure we chose the most appropriate meanings. If you don't do your due diligence, your results won't be valid. When Hegarty's team reran the analysis with this new database, their findings broadly agreed with the earlier farmer origin theory, locating the origin squarely in Anatolia about 8,000 years ago. From there, some branches of the language moved eastward and gave rise to languages including Persian and Hindustani. Other branches moved west to eventually develop into Greek and Albanian. But the analysis also recognizes the steppes as playing an important role as a secondary homeland for most European languages. After one branch traveled northward from Anatolia to the steppes, it radiated from there into northern Europe, giving birth to Germanic, Italic, Gaelic, and other European language families. Not convinced, mainstream historical linguists remain skeptical, however, of computational phylogenetics in general, and the new result in particular. The main criticism is that the approach relies mostly on vocabulary and ignores word sounds and structures, such as the stems, prefixes, and suffixes that make up a word. And the critics say that word meanings by themselves don't give enough information to draw firm conclusions, no matter how sophisticated the computation is. Thomas Olander, a historical linguist at the University of Copenhagen, says that the problem with depending on related words is that languages borrow words from one another all the time. Just seeing that there are words in common between two languages, then, doesn't mean the languages come from the same parent. The fact that English speakers now use the word sushi, for example, doesn't mean that English and Japanese are related languages. Instead, most linguists tend to trust sound shifts, such as the didwaido, to, zwei shift, along with similarities in the structures of words that can indicate which language they originated in. Word meanings can also be part of that mix, but they can't do it alone, Olander says. Hegarty's tree has other problems as well. For instance, it shows Celtic languages as being closely related to Germanic languages. But Olander says most historical linguists think Celtic languages are much more closely related to Italic languages. It's something that, again, is surprising, Olander says. I think surprising could be translated to it probably means that their method is wrong. Olander thinks it is far more likely that Celtic and Germanic branches coexisted closely for a long time and loaned one another words. An analysis based solely on shared word meanings shows them as more closely related than they actually are, he says. James Clarkson, a linguist at Cambridge University, also finds the early date for Proto-Indo-European and other details of the tree unconvincing, but he thinks computational phylogenetics is worth pursuing. And, if nothing else, he says, the most recent research created a very high-quality new dataset that will be important to historical linguists in general as they seek to solve many unsettled issues in their field. In the meantime, advocates of computational phylogenetics are likely to continue to promote their methods and seek legitimacy from the wider discipline. Hegarty thinks that as mainstream linguists get more comfortable with the method and the high-quality data it uses, they may give it more of a hearing. Clarkson, for one, says he's willing to be convinced. It's a developing field, and it's worth keeping an eye on, he says. Here's how Europeans quickly evolved lighter skin. Darker-skinned people lived in Europe until fairly recently, as Europeans spread imperialism around the world. They carried the genes for light skin with them. But even Europeans haven't been white for very long. New analysis of ancient European genes shows that other traits we associate with modern Europeans, such as tallness and the ability to digest milk, are also relatively recent additions to the continent's genetic profile. The new data, presented at the annual meeting of the American Association of Physical Anthropologists, comes from the genomes of 83 people found in archaeological sites across Europe, reports Anne Gibbons for Science. For years, researchers assumed that skin lightened as humans migrated from Africa and the Middle East into Europe about 40,000 years ago. A sun lower in the sky and shorter day lengths would have favored skin that more easily synthesized vitamin D. But researchers are now learning that other factors must have been at play. For example, earlier this year, the genome sequencing of a hunter-gatherer who lived in what is now Spain helped build the case that Europe was home to blue-eyed but dark-skinned people. This man, however, lived just 7,000 years ago. The researchers write that their analysis suggests that light skin was not yet widespread and ubiquitous in Europe at the time. Earlier work done with the genes of the 83 people in the new study, supported by linguistic evidence, also shows that populations in Europe about 8,000 years ago would have been mixed and diverse. The new study adds to this growing pile of evidence. Gibbons reports that the researchers found that Europeans probably couldn't have digested milk until about 4,300 years ago. And the story of skin pigmentation is complex. She writes, the new data confirm that about 8,500 years ago, early hunter-gatherers in Spain, Luxembourg, and Hungary also had darker skin. They lacked versions of two genes, SLC24A5 and SLC45A2, that lead to the pigmentation, and therefore pale skin in Europeans today. But in the far north, where low light levels would favor pale skin, the team found a different picture in hunter-gatherers. 7 people from the 7,700-year-old Motala archaeological site in southern Sweden had both light-skin gene variants, SLC24A5 and SLC45A2. They also had a third gene, HER2-OCA2, which causes blue eyes and may also contribute to light skin and blonde hair. Thus, ancient hunter-gatherers of the far north were already pale and blue-eyed, but those of Central and Southern Europe had darker skin. What we thought was a fairly simple picture of the emergence of depigmented skin in Europe is an exciting patchwork of selection as populations disperse into northern latitudes. Paleoanthropologist Nina Jablonski of Penn State told Science. This data is fun because it shows how much recent evolution has taken place. Ancient DNA and Neanderthals. DNA, deoxyribonucleic acid, is one of the best tools scientists have to learn about living things. It tells us who we are, where we come from, and what diseases we might be prone to. When studying evolution, DNA helps identify different species. However, DNA breaks down over time, so for most fossils, it's impossible to get any DNA. This makes it hard to answer questions about their looks, behavior, or population. But for some recently extinct species, scientists can still extract ancient DNA, aDNA, to learn more about their past. One such species is the Neanderthals, Homo neanderthalensis. Neanderthals were the first fossil hominins discovered, with fossils found in Belgium in 1829, but they weren't recognized as Neanderthals until later. The first fossils called Neanderthals were found in Germany in 1856, in the Neander Valley, where they got their name. Neanderthals split from modern humans about 500,000 years ago, likely evolving outside Africa, while humans stayed in Africa until about 100,000 years ago. Neanderthals adapted to cold climates in Europe and Asia, with features like short limbs and wide noses. Despite these differences, Neanderthals were closely related to humans and even lived in the same areas in Europe and the Middle East at the same time. Scientists use DNA to study the relationship between Neanderthals and modern humans, including questions like, did they interbreed? How much Neanderthal DNA do modern humans carry? What role do Neanderthal genes play in us today? By comparing human and Neanderthal genomes, scientists can answer these questions. But since DNA breaks down over time, it's hard to work with, making it a challenge to study ancient species like Neanderthals. DNA, the language of life. DNA, structure and function. DNA looks like a twisted ladder, with 2 long strands forming the sides and pairs of bases making up the rungs. The bases are adenine, A, thymine, T, guanine, G, and cytosine, C. Adenine always pairs with thymine, and guanine pairs with cytosine. For instance, if one side of the ladder reads AATG, the other side will read TTEC. This sequence of base pairs makes up our genetic code. DNA's main job is to control how proteins are made in our cells. This process has two steps: transcription and translation. First, an enzyme unwinds the DNA and makes a copy called messenger RNA, mRNA. This mRNA then moves to the ribosome in the cell's cytoplasm, where it is used to build a protein. Transfer RNA, tRNA, helps by bringing amino acids, which are linked together in the right order to form the protein. The mRNA and tRNA use groups of 3 bases, called codons, to specify which amino acids are needed. Sometimes a mutation changes a base in a codon, but if it doesn't alter the amino acid, like changing AA to AAG, both of which code for lysine, it's called a silent mutation. But if the mutation changes the codon to code for a different amino acid, it can affect the protein, which is known as a non-synonymous mutation. These mutations contribute to genetic diversity and evolution. DNA isn't all about coding for proteins. It includes exons, coding sequences, and introns, non-coding sequences. Exons make up only about 1% of our DNA and code for proteins, while introns were once considered junk DNA, but now are known to have important roles, such as regulating gene activity and maintaining chromosome structure. Most of our genetic material is in the nucleus of our cells, called nuclear DNA. This DNA is organized into 23 pairs of chromosomes, which separate during cell division. We inherit half of our nuclear DNA from each parent, with 1 pair of chromosomes, the sex chromosomes, determining our biological sex. For example, females typically have XX, and males have XY, though other combinations like XXY can occur. The Y chromosome, inherited only from fathers, helps trace paternal lineage. DNA is also found in mitochondria, the cell's energy producers. Mitochondrial DNA, mtDNA, is much smaller, containing about 37 genes. It is inherited solely from mothers and helps trace maternal lineage. While both Y chromosome DNA and mtDNA are useful for tracing deep ancestral lines due to their stability, they are much smaller than the nuclear genome. Nuclear DNA is in the cell nucleus and comes from both parents. It includes 23 pairs of chromosomes and determines most of our genetics. Mitochondrial DNA, mtDNA, is in the mitochondria, inherited from the mother, and helps trace maternal ancestry. Both types of DNA are used in genetic analysis, but have different applications due to their unique inheritance patterns. Challenges in extracting ancient DNA. DNA, the molecule that carries genetic information, breaks down over time, especially in organisms that have been dead for a long time. While it's easy to extract DNA from living organisms, finding usable DNA in fossils gets harder the further back in time we go. The oldest DNA ever recovered comes from mammoths that lived over 1 million years ago, found in frozen Siberian soil. However, the oldest DNA from early humans, like Neanderthals, is around 400,000 years old. This is because ancient human habitats were often warm and wet, which isn't ideal for preserving DNA. When scientists find ancient DNA, aDNA, they must handle it carefully to avoid contamination and preserve it as much as possible. The DNA they find is often fragmented and degraded, so they use a technique called polymerase chain reaction, PCR, to make many copies of the DNA for study. This helps scientists analyze and piece together the genetic information. Over time, DNA can also chemically change, which can lead to errors in reading the sequences. However, scientists can sometimes reverse these changes and use them to verify that the DNA is genuine. Extracting and analyzing aDNA is destructive, meaning it can damage the fossil. Therefore, scientists usually test extraction techniques on other fossils from the same site before working with valuable specimens. Fast facts. DNA preserves best in cold, dry environments. The oldest DNA recovered is over 1 million years old, but the oldest human DNA is about 400,000 years old. Extracting a DNA requires careful sampling and analysis, often destroying part of the fossil. Decoding the Neanderthal genome. Neanderthal mtDNA. The first analysis of any Neanderthal DNA was mitochondrial DNA, mtDNA, published in 1997. The sample was taken from the first Neanderthal fossil discovered, found in Feldhofer Cave in the Neander Valley in Germany. A small sample of bone was ground up to extract mtDNA, which was then replicated and analyzed. Researchers compared the Neanderthal mtDNA to modern human and chimpanzee mtDNA sequences and found that the Neanderthal mtDNA sequences were substantially different from both. Krings et al. 1997-1999. Most human sequences differ from each other by an average of 8.0 substitutions, while the human and chimpanzee sequences differ by about 55 co-substitutions. The Neanderthal and modern human sequences differed by approximately 27.2 substitutions. Using this mtDNA information, the last common ancestor of Neanderthals and modern humans dates to approximately 550,000 to 690,000 years ago, which is about 4 times older than the modern human mtDNA pool. Since this study was completed, many more samples of Neanderthal mtDNA have been replicated and studied. After successfully sequencing large amounts of mtDNA, a team led by Svante Pääbo from the Max Planck Institute reported the first complete mitochondrial DNA, mtDNA sequence for a Neanderthal. Green et al. 2008. The sample was taken from a 38,000-year-old Neanderthal from Vindija Cave, Croatia. The complete mtDNA sequence allowed researchers to compare this Neanderthal mtDNA to modern human mtDNA to see if any modern humans carried the mtDNA from a related group to the Neanderthal group. Later, Svante Pääbo's lab sequenced the entire mitochondrial genome of 5 more Neanderthals. Briggs et al. 2009. Sequences came from 2 individuals from the Neander Valley in Germany and 1 each from Mezmayskaya Cave in Russia, El Cidrón Cave in Spain, and Vindija Cave in Croatia. Though the Neanderthal samples came from a wide geographic area, the Neanderthal mtDNA sequences were not particularly genetically diverse. The most divergent Neanderthal sequence came from the Mezmayskaya Cave Neanderthal from Russia, which is the oldest and easternmost specimen. Further analysis and sampling of more individuals has led researchers to believe that this diversity was more closely related to age than it was to population-wide variance. Briggs et al. 2009. On average, Neanderthal mtDNA genomes differ from each other by 20.4 bases and are only 1/3 as diverse as modern humans. Briggs et al. 2009. The low diversity might signal a small population size. There is evidence that some other hominin contributed to the Neanderthal mtDNA gene pool around 270,000 years ago. Posch et al. 2017. A femur discovered in Germany had its mtDNA genotyped, and it was found that there was introgression from a non-Neanderthal African hominin, either Homo sapiens or closely related to us, around 270,000 years ago. This mitochondrial genome is also highly divergent from the Neanderthal average discussed previously, indicating that Neanderthals may have been much more genetically diverse in their more distant past. As for Neanderthal introgression into the modern human mtDNA genome, it is possible that the evidence of such admixture is obscured for a variety of reasons. Wang et al. 2013. Primary among these reasons is sample size. There are to date only a dozen or so Neanderthal mtDNA sequences that have been sampled. Because the current sample of Neanderthal mtDNA is so small, it is possible that researchers simply have not yet found the mtDNA in Neanderthals that corresponds to that of modern humans. Neanderthal nuclear DNA. There have been many efforts to sequence Neanderthal nuclear genes, with an eventual goal to sequence as much of the Neanderthal genome as possible. In 2014, the complete genome of a Neanderthal from the Altai Mountains in Siberia was published. Prufer et al., 2014. This female individual's genome showed that her parents were likely half-siblings, and that her genetic line showed evidence of high rates of incestuous pairings. It is unclear whether this is due to her living in a small and isolated population, or if other factors may have influenced the lineage's inbreeding. Their analysis also showed that this individual was closely related to both modern humans and the Denisovans, another ancient human population. By their analysis, there was only a very small margin by which Neanderthal and Denisovan DNA differed exclusively from modern humans. Fast facts. Neanderthals are genetically distinct from modern humans, but are more closely related to us than chimpanzees are. The Neanderthal and modern human lineages diverged about 550,000 years ago. So far, we have no evidence of Neanderthal mtDNA lineages in modern humans. Neanderthals were not as genetically diverse as modern humans were at the same period, indicating that Neanderthals had a smaller population size. Neanderthal nuclear DNA shows further evidence of small population sizes, including genetic evidence of incest. As technology improves, researchers are able to detect and analyze older and more fragmentary samples of DNA. Another lost relative, identifying the Denisovans. Who were the Denisovans? Scientists have also found DNA from another extinct hominin population, the Denisovans. The first remains of the species found were a single fragment of a phalanx, finger bone, and 2 teeth, all of which date back to about 40,000 years ago. Reich et al., 2010. Since then, a Denisovan mandible, or lower jaw, has been found in Tibet, Chen et al., 2019, and a Denisovan molar has been found in Laos, Demeter et al., 2022. Other fossil hominins, such as the Homo longi, remains from northern China, G et al., 2021, and the Dali cranium from northwestern China may belong to the Denisovans. But without comparable fossils and genetics, it is difficult to know for sure. This species is the first fossil hominin identified as a new species based on its DNA alone. Denisovans are close relatives of both modern humans and Neanderthals, and likely diverged from these lineages around 300,000 to 400,000 years ago. They are more closely related to Neanderthals than to modern humans. You might be wondering, if we have the DNA of Denisovans, why can't we compare them to modern humans like we do Neanderthals? Why isn't this article about them too? The answer is simply that we don't have enough DNA and fossils to make a comparison. The single-digit specimen pool of Denisovans found to date is statistically far too small a dataset to derive any meaningful comparisons. Until we find more Denisovan material, we cannot begin to understand their full genome in the way that we can study Neanderthals. The lack of more and more morphologically diagnostic Denisovan fossils is the reason why scientists have not yet given them a species name. Denisovans are known from only a few isolated fossils, but are the first hominins identified as a new species on a genetic basis. There are not enough Denisovan fossils and genomes to have as clear a picture of their species as we do Neanderthals. Evidence for interbreeding. Shared DNA. What does it mean? Homo sapiens and Homo neanderthalensis are different species, yet you are reading this webpage about them potentially interbreeding with each other. So, what does that mean exactly? Modern humans and Neanderthals lived in separate regions, evolving along separate evolutionary lineages for hundreds of thousands of years. Even so, Neanderthals are still our closest currently known relative. Because of that evolutionary proximity, despite being recognized as different species, it is still possible that members of our two species exchanged genetic information. This exchange of DNA is called introgression or interbreeding. When looking for evidence of interbreeding, scientists do not search billions and billions of base pairs. Instead, there are specific regions of the genomes that are known to be highly variable in modern humans, along with several million single nucleotide polymorphisms, SNPs, where the given base at a single location can vary among people. The difference between the total genome and these specific regions, sites, can lead to some confusion. In terms of the total genome, humans and chimpanzees are 98 and 99% similar. Yet, it is possible for individuals to have up to 4% Neanderthal DNA. That difference is accounted for in that 4% of the highly variable genome is inherited from a Neanderthal source, not 4% of the entire genome. If one was to look at the modern human genome as a whole, at least 98–99% is the same, inherited from our common ancestor with Neanderthals. Neanderthal Homo sapiens, interbreeding. Neanderthals are known to contribute up to 1 to 4% of the genomes of non-African modern humans, depending on what region of the world your ancestors come from. And modern humans who lived about 40,000 years ago have been found to have up to 6–9% Neanderthal DNA. Fu et al., 2015. Because Neanderthals likely evolved outside of Africa, no Neanderthal fossils have been found in Africa to date. It was thought that there would be no trace of Neanderthal DNA in African modern humans. However, a study in 2020 demonstrated that there is Neanderthal DNA in all African Homo sapiens. Chen et al. 2020. This is a good indicator of how human migration out of Africa worked. That Homo sapiens did not leave Africa in one or more major dispersals, but that there was gene flow back and forth over time that brought Neanderthal DNA into Africa. The evidence we have of Neanderthal modern human interbreeding sheds light on the expansion of modern humans out of Africa. These new discoveries refute many previous hypotheses in which anatomically modern humans replaced archaic hominins, like Neanderthals, without any interbreeding. However, even with some interbreeding between modern humans and now extinct hominins, most of our genome still derives from Africa. For many years, the only evidence of human Neanderthal hybridization existed within modern human genes. However, in 2016, researchers published a new set of Neanderthal DNA sequences from Altai Cave in Siberia, as well as from Spain and Croatia, that show evidence of human Neanderthal interbreeding as far back as 100,000 years ago, farther back than many previous estimates of humans' migration out of Africa. Kulwilm et al. 2016. Their findings are the first to show human gene flow into the Neanderthal genome, as opposed to Neanderthal DNA into the human genome. These data tells us that not only were human Neanderthal interbreeding events more frequent than previously thought, but also that an early migration of humans did in fact leave Africa before the population that survived and gave rise to all contemporary non-African modern humans. We previously mentioned the lack of genetic contributions by Neanderthals into the modern human mtDNA gene pool. As we have shown that Neanderthal human interbreeding did occur, why wouldn't we find their DNA in our mtDNA as well as our nuclear DNA? There are several potential explanations for this. It is possible that there were at one point modern humans who possessed the Neanderthal mtDNA, but that their lineages died out. It is also highly possible that Neanderthals did not contribute to the mtDNA genome by virtue of the nature of human-Neanderthal admixture. While we know that humans and Neanderthals bred, we have no way of knowing what the possible social or cultural context for such breeding would have been. Because mtDNA is passed down exclusively from mother to offspring, if Neanderthal males were the only ones contributing to the human genome, their contributions would not be present in the mtDNA line. It is also possible that while interbreeding between Neanderthal males and human females could have produced fertile offspring, interbreeding between Neanderthal females and modern human males might not have produced fertile offspring, which would mean that the Neanderthal mtDNA could not be passed down. Finally, it is possible that modern humans do carry at least one mtDNA lineage that Neanderthals contributed to our genome, but that we have not yet sequenced that lineage in either modern humans or in Neanderthals. Any of these explanations could underlie the lack of Neanderthal mtDNA in modern human populations. Human Denisovan and Neanderthal Denisovan interbreeding. Given that scientists have DNA evidence of another hominin species, the Denisovans, is there any evidence for interbreeding among all three species? Yes. Comparison of the Denisovan genome to various modern human populations shows up to 4 to 6% contribution from Denisovans in non-African modern human populations. This concentration is highest in people from Papua New Guinea and Oceania. It makes sense that interbreeding would appear in these Southeast Asian and Pacific Island communities, as their ancestors migrated from mainland Asia, where Denisovan fossils have been found. There is also substantial evidence for Denisovan Neanderthal interbreeding, including one juvenile female that appears to be a fifth-generation hybrid of a Neanderthal female parent and Denisovan male parent, Sloan et al., 2018. Finding more Denisovan fossils will hopefully mean developing a more complete picture of Denisovan genetics so that scientists can explore these interactions in more detail. Fast facts. Neanderthals have contributed 1.4% of the DNA of humans of Eurasian descent. Neanderthals have also indirectly contributed to the genome of modern humans of African descent via ancient modern human migrations back into Africa. There are multiple possible explanations as to why there is not Neanderthal mtDNA in the modern human gene pool. Denisovans also contributed up to 4 to 6% of the genome of modern humans in certain regions and interbred with Neanderthals as well. DNA genotypes and phenotypes. What do Neanderthals genes do? Genes and evolution. Besu. While much of the genetic diversity discussed above came from inactive, non-coding, or otherwise evolutionarily neutral segments of the genome, there are many sites that show clear evidence of selective pressure on the variations between modern humans and Neanderthals. Researchers found 78 loci at which Neanderthals had an ancestral state, and modern humans had a newer, derived state. Green et al., 2010. 5 of these genes had more than one sequence change that affected the protein structure. These proteins include SPAG17, which is involved in the movement of sperm, PCD16, which may be involved in wound healing, TTF1, which is involved in ribosomal gene transcription, and RPTN, which is found in the skin, hair, and sweat glands. Other changes may not alter the sequence of the gene itself, but alter the factors that control that gene's replication in the cell, changing its expression secondarily. This tells us that these traits were selected for in the evolution of modern humans and were possibly selected against in Neanderthals. Though some of the genomic areas that may have been positively selected for. In modern, humans may have coded for structural or regulatory regions. Others may have been associated with energy metabolism, cognitive development, and the morphology of the head and upper body. These are just a few of the areas where we have non-genetic evidence of differentiation between modern humans and Neanderthals. While the study of DNA reveals aspects of relatedness and lineage, its primary function is, of course, to control the production of proteins that regulate an organism's biology. Each gene may have a variety of genotypes, which are the variances that can occur within the site of a particular gene. Each genotype codes for a respective phenotype, which is the physical expression of that gene. When we study Neanderthal DNA, we can examine the genotypes at loci of known function and can infer what phenotype the Neanderthal's mutations may have expressed in life. Below, explore several examples of Neanderthal genes and the possible phenotypes that they would have displayed. Red-headed, pale-skinned Neanderthals. Ancient DNA has been used to reconstruct aspects of Neanderthal appearance. A fragment of the gene for the melanocortin-1 receptor, MRC1, was sequenced using DNA from two Neanderthal specimens from Spain and Italy, El Cidron, 1252, and Montelesini, La Luisa Fox, et al. 2007. MC1R is a receptor gene that controls the production of melanin, the protein responsible for pigmentation of the hair and skin. Neanderthals had a mutation in this receptor gene which changed an amino acid, making the resulting protein less efficient and likely creating a phenotype of red hair and pale skin. The reconstruction below of a male Neanderthal by John Gertcher features pale skin but not red hair. How do we know what this phenotype would have looked like? Modern humans display similar mutations of MC1R, and people who have two copies of this mutation have red hair and pale skin. However, no modern human has the exact mutation that Neanderthals had, which means that both Neanderthals and humans evolved this phenotype independent of each other. If modern humans and Neanderthals living in Europe at the same time period both evolved this reduction of pigmentation, it is likely that there was an advantage to this trait. One hypothesis to explain this adaptation's advantage involves the production of vitamin D. Our bodies primarily synthesize our supply of vitamin D rather than relying on vitamin D from food sources. Vitamin D is synthesized when the sun's UV rays penetrate our skin. Darker skin makes it harder for sunlight to penetrate the outermost layers and stimulate the production of vitamin D. And while people living in areas of high sun exposure will still get plenty of vitamin D, people who live far from the equator are not exposed to as much sunlight and need to optimize their exposure to the sun. Therefore, it would be beneficial for populations in colder climates to have paler skin so that they can create enough vitamin D, even with less sun exposure. Neanderthals, language, and FOXP2. The FOXP2 gene is involved in speech and language, Lai et al. 2001. Mutations in the FOXP2 gene sequence in modern humans led to problems with speech and oral and facial muscle control. The human FOXP2 gene is on a haplotype that was subject to a strong selective sweep. A haplotype is a set of alleles that are inherited together on the same chromosome, and a selective sweep is a reduction or elimination of variation among the nucleotides near a particular DNA mutation. Modern humans and Neanderthals share two changes in FOXP2, compared with the sequence in chimpanzees. Krauss et al., 2007. How did this FOXP2 variant come to be found in both Neanderthals and modern humans? One scenario is that it could have been transferred between species via gene flow. Another possibility is that the derived FOXP2 was present in the ancestor of both modern humans and Neanderthals, and that the gene was so heavily favored that it proliferated in both populations. A third scenario, which the authors think is most likely, is that the changes and selective sweep occurred before the divergence between the populations. While it can be tempting to infer that the presence of the same haplotype in Neanderthals and humans means that Neanderthals had similar complex language capabilities. There is not yet enough evidence for such a conclusion. Neanderthals may also have their own unique derived characteristics in the FOXP2 gene that were not tested for in this study. Genes are just one factor of many in the development of language. ABO blood types and Neanderthals. The gene that produces the ABO blood system is polymorphic in humans, meaning that there are more than 2 possible expressions of this gene. The genes for both A and B blood types are dominant, and O type is recessive, meaning that people who are type A or B can have genotypes of either AA or AO, or BB and BO, and still be A or B blood type. But to have type O blood, one must have a genotype of OO. Various selection factors may favor different alleles, leading to the maintenance of distinct blood groups in modern human populations. Though chimpanzees also have different blood groups, they are not the same as human blood types. While the mutation that causes the human B blood group arose around 3.5 MA, the O group mutation dates to around 1.15 MA. When scientists tested whether Neanderthals had the O blood group, they found that 2 Neanderthal specimens from Spain probably had the O blood type, though there is the possibility that they were OA or OB, Lalueza-Fox et al. 2008. Though the O allele was likely to have already appeared before the split between humans and Neanderthals, it could also have arisen in the Neanderthal genome via gene flow from modern humans. Bitter taste, perception, and Neanderthals. The ability to taste bitter substances is controlled by a gene, TAS-R38. Some individuals are able to taste bitter substances, while others have a different version of the gene that does not allow them to taste bitter foods. Possession of 2 copies of the positive tasting allele gives the individual greater perception of bitter tastes than the heterozygous state in which individuals have 1 tasting allele and 1 non-tasting allele. 2 copies of a non-tasting allele leads to inability to taste bitter substances. When scientists sequenced the DNA of a Neanderthal from El Cidrón, Spain, for the TAS23R38 gene, they found that this individual was heterozygous and thus was able to perceive bitter taste, although not as strongly as a homozygous individual. With 2 copies of the tasting allele would be able to. Lalueza-Fox et al. 2009. Both of these haplotypes are still present in modern people, and since the Neanderthal sequenced was heterozygous, the 2 alleles, tasting and non-tasting, were probably both present in the common ancestor of Neanderthals and modern humans. Though chimpanzees also vary in their ability to taste bitterness, their abilities are controlled by different alleles than those found in humans, indicating that non-tasting alleles evolved separately in the hominin lineage. Microcephaline and archaic hominins. The microcephaline gene relates to brain size during development. A mutation in the microcephaline gene, MCPH1, is a common cause of microcephaly. Mutations in microcephaline cause the brain to be 3 to 4 times smaller in size. A variant of MCPH1, haplogroup D, may have been positively selected for in modern humans, and may also have come from an interbreeding event with an archaic population. Evans et al. 2006. All of the haplogroup D variants come from a single copy that appeared in modern humans around 37,000 years ago. However, haplogroup D itself came from a lineage that had diverged from the lineage that led to modern humans around 1.1 million years ago. Although there was speculation that the Neanderthals were the source of the microcephaline haplogroup D, Evans et al. 2006, Neanderthal DNA sequenced does not contain the microcephaline haplogroup D, Green et al. 2010. Enamel formation and dental morphology. While changes to the genome can directly affect the phenotypes displayed in an organism, altering the timing mechanism of protein production can cause very similar effects. MicroRNA, miRNA, is one such mechanism. A cell uses miRNA, 'miRNA, 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 One miRNA can target multiple genes by binding its seed region to messenger RNA that would otherwise have carried that information to the ribosome to be transcribed into proteins, preventing transcription from taking place. In hominins, one particular miRNA called MIR1304 is exhibited in both an ancestral and derived condition. The derived condition has a mutation at the seed region, which allows it to target more mRNA segments but less effectively. This means that in the derived state, some genes will be more strongly expressed due to a lack of suppression. One such trait is the production of enamelin and amylotin proteins, both used in dental formation during development. The suppression of production in Neanderthals and subsequent lack of suppression in modern humans could be a contributing factor to some of the morphological differences between Neanderthal and modern human dentition. Immune response. Research shows that Neanderthal DNA has contributed to our immune systems today. A study of the human genome found a surprising incursion of Neanderthal DNA into the modern human genome, specifically within the region that codes for our immune response to pathogens. Daneman et al. 2016. These particular Neanderthal genes would have been useful for the modern humans arriving in Europe, whose immune systems had never encountered the pathogen within Europe and would be vulnerable to them, unlike the Neanderthals, who had built up generations of resistance against these diseases. When humans and Neanderthals interbred, they passed this genetic resistance to diseases onto their offspring, allowing them a better chance at survival than those without this additional resistance to disease. The evidence of this genetic resistance shows that there have been at least 3 incursions of non-human DNA into the genes for immune response, 2 coming from Neanderthals and 1 from our poorly understood evolutionary cousins, the Denisovans. Clotting, depression, and allergies. While many of the genes that we retain for generations are either beneficial or neutral, there are some that have become deleterious in our new, modern lives. There are several genes that our Neanderthal relatives have contributed to our genome that were once beneficial in the past, but can now cause health-related problems. Simonti et al. 2016. One of these genes allows our blood to coagulate, or clot, quickly, a useful adaptation in creatures who often injure while hunting. However, in modern people who live longer lives, this same trait of quick clotting blood can cause harmful blood clots to form in the body later in life. Researchers found another gene that can cause depression and other neurological disorders, and is triggered by disturbances in circadian rhythms. Since it is unlikely that Neanderthals experienced such disturbances to their natural sleep cycles, they may never have expressed this gene. But in modern humans who can control our climate and for whom our lifestyle often disrupts our circadian rhythms, this gene is expressed more frequently.
Join the Compuverse Patreon for exclusive content, behind-the-scenes access, and special perks. Support the channel and connect with a community of revolutionary intellectuals and scholars. Click the link in the description to join now.